Hello everyone, welcome back to Microbiology Solutions. Today we are going to discuss about Widal test. So Widal test is a agglutination test or tube agglutination test done to detect salmonella infection or typhoid fever, which is an acute, life-threatening and fever-causing illness which is transmitted by the ingestion of contaminated food or water with the feces of a person who is infected with the bacteria Salmonella typhi or Salmonella paratyphi A, B and C respectively. And antibodies against this organism will appear in human body after 2 to 3 weeks of infection. And this test measures agglutinating antibodies directed against Salmonella O somatic surface antigen or Salmonella H flagellar antigen of the suspected organism. So, Salmonella possesses somatic O antigen and flagellar H antigen. Widal test basically measures or look for the presence of agglutinating antibodies against O antigen and H antigens produced by Salmonella. The Widal test detects antibodies against O and H antigens. The type of techniques is used here is direct agglutination, which was developed by Fernand Widal in 1896. So moving on to the symptoms of typhoid fever when we have to go for a Widal test. What are the indications to go for a Widal test are high temperature, the patient is feeling weak, headache, gastroenteritis and diarrhea. These are the common symptoms of having a typhoid fever or an enteric fever. The principle of Widal test is that antibody in the serum which is produced in response to the infection caused by Salmonella and the kit contains antigen suspensions which are killed bacterial suspensions and they were stained. These bacterial suspensions were stained to enhance the reading of agglutination test. If we look for the agglutination with the naked eye, the visibility may not be proper, so that the antigen suspensions in the kit is stained with certain dyes. The blue stained antigens are specific to the somatic antigen or O antigen and the red or pink stained antigens are specific to the flagellar antigen or H antigens. So the reagent blue particular for O antigen and red is particular for H antigen. So typically the patient serum is diluted into a series of tubes or we do a serial dilution with the patient serum using normal saline or physiological salines on different tubes or wells on a slide, which will then made react with the bacterial specific antigen for the suspected disease. And the test measures agglutinating antibodies or the clumping of antibodies directed against Salmonella O somatic antigen or H flagellar antigens of Salmonella. The controls of Widal test, the Widal negative control which contains no antibodies against specific bacteria, and Widal positive control which contains a standardized goat antiserum with polyspecific antibodies having specific reactivity towards S. typhi O and H antigens, S. paratyphi AH and BH, S. paratyphi AO and BO, S. paratyphi CO and CH antigens, which is useful in the validation of the performance of Widal reagent. So when we are doing a test, we should run a positive and a negative control parallelly so that we can say that our result is 100% correct. And a positive and negative control should be run with each test, and if the reagent fails to agglutinate with the positive control or does agglutinate with the negative control, it should be discarded. You should take utmost care to run the test along with positive as well as negative control. So this rapid slide test or a screening test for Widal, which is done on a slide. The procedure is you can place one drop or you can even transfer using micropipette 50 microliter of the serum of a patient into a circle in the slide and add one drop of well shook antigens OH, BO and BH or AH and BH respectively into each circle. The antigen you should be selecting should be appropriate to the endemicity in your own area. Which strain of salmonella is endemic in your area? So pertaining to that, you should look for the antigen. If paratyphi A infection is more, you should always look for paratyphi A or AH antigens. And spread the contents to fill the whole circle area. Mix and rotate the slide for 1 minute and you can observe for agglutination. If there are agglutination, you have to do a tube agglutination test. You can also report it as slide or rapid agglutination test positive. But in order to confirm the test, you should go for a tube agglutination of salmonella. So you should go for a confirmatory test using a Widal reagent. And the interpretation of salmonella infection, salmonella typhi O positive means recent infection because it is a somatic antigen which represents a recent infection. Salmonella typhi H antigen if it is giving positive, which means an old infection which gives positive to the flagellar antigen. Salmonella paratyphi B positive means it could be a carrier who can infect others. Also, patient history should be taken into account before giving the result. So this is about rapid screening test for Widal. So in next class we would check for the tube agglutination test for Widal, which is more important in the second week of salmonella infections. So when we suspect a salmonella infection, at the first week of infestation we would go for a blood culture test, and second week we usually go for a Widal test. It could be a slide agglutination or it can be a tube agglutination, and the third week as the disease subsides we would go for a urine as well as feces or stool culture. This is about today's class. If you have any doubts, please feel free to contact. Please do like, share and subscribe to our channel. Thank you.